So next we have Dr. Yellen. He's from the engineering school, the Department of Mechanical and Engineering. Ben has become recently interested in using his methods of magnetics and applying it to the single cell field and single cell immunology. So he's going to talk today about his random access single cell chip for the study of HIV latency.
Okay. Which one is this? Thanks very much for the introduction. I'm very pleased to be talking in this session for people who don't know anything about AIDS. And I'll take that one step further. I know very little about medicine as well. I come from the field of engineering, and my expertise is on using magnetic and electric fields to manipulate biological materials, particles, cells. Not really cells initially, but then I met Dave Murdoch, who is very interested in single cell, and he's a very excitable guy too and likes new technologies. So we started talking, and it started a long dialogue over a year of just figuring out why is another single cell tool necessary. So this is the pitfall of being an engineer, is that if you have a hammer, you see everything as a nail. And so we were trying to figure out really why we should focus on developing a very long project on a new single cell tool. And it turns out that there are some good reasons, and the existing tools out there are not good enough, particularly in the area where we would like to observe a cell over very long periods and then take that cell and do DNA analysis or RNA analysis on it afterwards. In fact, what we decided is that we would like our single cell apparatus to function very much like a computer memory chip, where we could write cells to an array and then read the cells repeatedly looking at fluorescence or other states, and then extract a single cell from the array, so writing a 1 to a 0 or 0 to a 1 and then doing RNA analysis on it. So that's the big picture. It turns out that HIV is one of these areas where we do need to understand single cells and watch them for a long period before we decide to extract them. And I don't think I need to go into too much detail here, but I will give you my view of what's going on as a barrier to an HIV cure. Right now we have what is considered to be a functional cure for HIV, where we inject people with antiretroviral therapies or they take it orally. And this activates the immune system and it kills any cells that are actively producing HIV virus. But there are also a very small subset of cells that remain immunologically silent. They, in all practical purposes, cannot be recognized by the immune system to be killed. But that also represents a problem because we can't study them, because we have no way to tag them and to isolate them and to study them either in bulk population or at the single cell level. So one of the barriers to this cure is how do we try to wake these cells up from their latency to where the immune system can recognize them and destroy them. So in the field of HIV latency and activation, there are some good cell models that exist out there, particularly the J-lat lines, several of these lines, which are engineered to be latent. And they, in many cases, have reporter genes engineered into them so that when they come out of latency, they will change color by either putting a GFP marker in them. But the problem here is that these behave very different to cells in vivo. And even among the cell lines, there's a large variability in how they respond to different drugs. So this is the J-lat 8.4 line, and this is the J-lat 11.4 line. And what we see here is the percentage of which are activated upon exposure to this set of drugs. And we see that some of them appear to be activated by common drugs among both of these lines, but some of them are selectively activated. And this is not something that we can even study in vivo yet. So this is what is motivating some of the single cell tools out there, because we would like to get rid of, to really create a reductionist experiment where the cells in a controlled environment, we can put uniform drugs onto single cells and then observe at the single cell level without interference from other cells how they come out of latency, what percentage comes out, and potentially also to look at their genome immediately after they come out of latency. So I'm going to now give an overview of the single cell tools that currently exist, and then I'll describe our tool and why there are some reasons that it can have some applications that other tools cannot do. So the first example is from the Chris Love group at MIT, and they are basically creating a morphological template of wells. So you see the wells right here, and they essentially just let cells passively settle into these wells, and it settles by a random Poisson distribution. Some wells have 0, 1, or 2 of the single cells in each of the wells. And then they can do certain things like put cytokine capture antibodies and look at secretion of these cells, and that's what you see here. And they're also developing tools where you could take a robot and extract single cells from the wells of interest and look at their genome. But you may imagine that the population efficiency is not very good, especially if you want to create single cell pairs. And additionally, it's very cumbersome to remove cells from each of these individual wells. So they already made some good breakthroughs, but there's a lot of room for improvement here. We all have recently obtained a Fluidigm instrument. So this is in the IGSP, I believe, second floor of CMOS. And this is a very nice tool for analyzing the genome and doing transcriptome analysis on arrays of single cells. This operates by using fluidic forces, and it traps single cells into little wells here. I guess in this array right here, this is one of the fluidic traps. And after that, they can lyse it and look at, do PCR on it and look at up to 69 genes, or 96 genes and 96 single cells. The problem here is that they can't culture the cells for very long periods because of nutrient deprivation and other issues. They basically can just introduce the cells into the array and then immediately do the genome analysis. So they have one picture of what's happening right before they do the genome analysis, and they don't have the ability to watch and then only selectively perform single cell sequencing of the ones that are just coming out of latency, for example. There's another tool that is more active and programmable. This is called this is from Silicon Biosystems. It uses electric fields, in particular dielectrophoresis, which is DEP. And it creates these cages of electric field that trap single cells and can move them around. And so in principle, this is a good tool for coordinating and separating cells, and is commonly used in doing cancer cell therapy and isolating circulating tumor cells. The problem is that due to the large number of electrodes here, it is very difficult to manipulate more than 10 or 20 cells. So since the latency problem is a very rare event, something like 1 in a million, it just doesn't have the scale or programmability to ever get to the point where we can look at latent cells, particularly in vivo. There's another recent work by the Weinberger group on using these fingering areas right here to capture latent single cells, and they can watch them over long periods of time and see, changes to their morphology and interaction with cytokines. The problem is that it's very difficult to retrieve cells from these fingers. And so these are essentially the main single-cell tools that are out there. And this provides motivation for using a different type of approach to organize single cells into large arrays that are based on circuit theory. So we use magnetic circuits that have scalability. They can control or operate on up to 1 million cells potentially with large enough areas. And it's comprehensive, so we're developing systems where we can culture the cells for up to a week while watching them. Its magnetic materials tend to be very compatible with biological materials, so they're relatively non-invasive. And we're going to look at some of the gene regulators that occur upon binding of cells to magnetic nanoparticles, but from what we have seen so far, they look to be relatively non-invasive. One of the very nice features of this magnetic system is that we can organize single-cell pairs and study the interactions for, in particular, the kill approach and what is commonly known as the shock and kill approach for waking up cells out of latency and then studying how the CD8s come and kill them. And finally, flexibility. If we want to, we can program the distance between single-cell pairs and maybe cause them to touch and analyze the effects of the cell-cell communication based on the length of duration of them touching or the distance that they're maintained apart from each other. So this is the motivation. And now I want to play a few movies that shows you how this system works. So basically we are using from circuit theory the same ideas that are used in normal circuits. So we are developing conductors and capacitors and transistors and diodes. And these are the elements of an electrical circuit, but to move electrons. Here we're doing the same thing, but moving matter instead. So I'm going to show you that initially here we have the first elementary magnetic particle conductor. And what you see here is a magnetic pattern that we patterned by photolithography. And when we apply a rotating field, so that's what you see right here, this is the direction of the field, it causes the magnetic beads, in this case, to move from one magnet curved surface to the next. And the speed at which these magnetic particles move across the substrate is linear with the frequency. So we have essentially the equivalent of Ohm's law, but for magnetic particle transport, where omega is the rotation frequency, and I is the current, and R has to do with the geometry that is similar to the resistance of a circuit. When we apply the rotating field in the clockwise sense, the bead moves right. When we apply a counterclockwise field, the bead moves left. So it behaves also in positive and negative sense, the same way that Ohm's law V equal IR behaves. So the next circuit element that we need to develop is a diode. So this is something that allows for one-way electrical current flow. Here we show that by combining this junction into a T pattern, when we apply a counterclockwise rotating field, the magnetic bead can proceed in the positive X direction. But when we have a counterclockwise rotating field, the bead cannot move in the negative X direction. It actually moves in a different direction. So this is an example of an asymmetric current conductor or a rectifier. This is what a diode is. And here is a plot of the velocity versus frequency. So if the frequency is in the negative sense, there is no bead transport. If it's in the positive sense, it moves right. So now that we have the ability to control the direction of particle motion, the next thing we want to do is develop sites that can trap particles and store them in an array. So these are our memory elements. And we basically combine this diode function with a magnetic track. So this diode closes in on itself such that if we apply a positive clockwise rotating field, the bead can get into this diode. And if we apply a negative rotating field, counterclockwise rotating field, the bead cannot escape the diode because this is a reverse bias configuration. So just to give you indication that this works on cells, this is an example of a single cell that has been transported and trapped locally in our capacitor. This is an immune cell. I think it's actually a mouse cell. And we're repeating these experiments here right now with human T cells, CD4 cells. This is just a fluorescence image of a movie of what's going on and shows that we indeed are trapping single cells. So all I've shown you are passive circuits. So these act globally. I'm applying one field and the structure of the magnetic tracks automatically guides many thousands or millions of cells in parallel to their locations. But in order to select one out of these many millions, we need active circuits. We need transistors. So this is the first example of a transistor where we combine a current line. So we pass current through this line here with this magnetic track so that we can turn on and turn off the ability of a bead to jump across a junction. So this system behaves very similarly and almost analogously to a semiconductor, we can consider this gap to be semi-conducting to the bead. And only when this current line is turned on, the gate electrode, the bead can jump across the gap. And when it's turned off, it cannot jump across the gap. So you just saw there the first bead did not jump across and the second bead did. So what we're able to do now is to say, you can go down row 1, and where you cannot go down row 2. The problem that we had initially is that it takes a lot of current to do this. So we've started performing some optimization studies. I don't want to go into the details here, but what we're trying to do is to lower the power of the switching so that we can switch hundreds of these channels at the same time. And now we can get from 100 milliamps to potentially 1 to 10 milliamps of switching magnitude. We need actually two types of transistors. One is to tell it which row to go down, and another is to get it out of an apartment. So this is the T-junction that I showed you originally, where we now overlay a diagonal current line. And the purpose of this current line is to allow us to turn on the current line and get the bead out of the apartment. That will then allow it to be sequenced. So in this example, the first bead we did not turn the current on, the second bead we did, it jumps out, and the third bead we didn't. So now that we have all of these fundamental circuit elements, we want to put them all together to make a multiplexed array. This is just an example of a 3 by 3 array where we have columns A, B, and C, and rows 1, 2, and 3. And by turning on and turning off the word line and the bit line at the appropriate times, we can populate an arbitrary pattern in the array. So you can see where we're trying to go here. Again, we're doing this with magnetic beads, but we're shortly going to be transitioning to cells. So this current line is off, this current line is off, but the third row is on, so the bead will move down the top row. And then we turn on the first column, and now the bead ends up in the A1 apartment. After that, we turn this one off, and we turn on the C column, and then the second bead ends up into the C1 apartment. And so by repeating this process, we show that we can create an arbitrary pattern. In this case, it turns out that we're going to make an X pattern, but eventually we want to be able to populate a full array, and then once we have this array, start to do our immunological studies of exposing them to drugs at different combinations or sequentially of the same drug and observe which ones comes out of latency and then extract those cells from the array. So some of the conclusions here that we have so far is that magnetic circuits are a pretty good tool for coordinating the assembly of large numbers of single cells and particles. And they require, in order to do massively parallel manipulation, a series of conductors, diodes, capacitors, transistors, and multiplexers to make it really act like a memory. So this is what we have done so far, and our scaling trends suggest that we can manipulate up to about 1 million cells or 100,000 cells per square inch of chip space. However, what we still need to do is to develop the surrounding instrumentation that makes this system biologically relevant. So this is what I proposed in the CNIHR grant, is number one is to develop, specific aim one is to develop a automated circuitry for long-term study of single cell arrays, which is essentially scales our 3 by 3 array up to one that is capable of arraying 10,000 or 1,000 cells. And we want to also automate the process so that I push one button and it tells the cell exactly where to go. We also need to develop a microincubator that allows these cells to maintain viability for extended periods. And then once we have this large array, we want to start studying what happens to these cells following stimulation with an anti-latency compound. So the one we have selected is vorinostat, and we are going to study the latency activation process of 2 infected cell lines. One's the JLAT5A8 and one is the JLAT 11.1 that are occurring under various doses of vorinostat. And we're going to then analyze the GFP positive and GFP negative cells by transcriptome analysis post-stimulation. And what this is allowing us to do is to understand which genes are being turned on and turned off immediately after latency activation. So this is one of the things that will allow us to eventually develop better drugs to interfere or promote these these epigenetic events that occur in latency activation. So our methodology is to use these magnetic circuits at every 4-hour intervals. We are going to pull one of the rows of the or just a set of GFP positive, GFP negative cells. So we're starting off with an 8x8 array here, and you can see essentially what the circuit looks like. Here we are developing the microincubator. So this is the rotating field setup, and we're using 3D printer to heat the chip, to create a substrate to hold the fluid chamber and resistive heater. Something wrong? Sorry. And we're also going to expose it to gas, and so we're trying to build the entire incubator so that it sits under a microscope and maintains viability of cells for up to a period of a week. There are potential challenges to this approach, so we're still at the very early stages, and I just want to give you a flavor of what we're trying to do. First of all, as many people note, that this is an in vivo or ex vivo system, so that this extracellular environment may cause problems for the single cells and induce non-native response. Another issue that has come up is that magnetic labeling may interfere with HIV expression, so we're going to study this as a separate system. People have also commented that maybe the expression kinetics occurs too slowly for us to really analyze what's happening in single cell analysis. So maybe the GFP buildup in a single cell takes 12 hours, and that's way longer after it has already come out of latency. So one way around this is to just analyze all the cells that come out and try to pair that with the fluorescence expression at different time intervals. And finally, if we ever want to get to a clinical relevancy, we have to find and study these in vivo latent cells which don't have a marker that allows us to know they've come out of latency. So this is the major problem with trying to study in vivo cells, latent cells in vivo, is that there's no way to tell the difference. There's no way to label them selectively and pull them out, and this is because the immune system can't recognize them, and therefore we can't at present. Maybe there will be some new tools that emerge that allows us to specifically label these immune latent cells in vivo or taking them out ex vivo, but so far I do not know of any tools that can do this. So this is just a summary slide of the impact and future directions. We are trying to develop a single-cell platform. This is not just relevant to HIV latency, but also in other fields of potentially cancer and other immune response. The coupling between observing time-resolved single-cell imaging with genome expression provides a powerful tool for getting big data of single cells. In particular, we are interested not only in just looking at single-cell arrays, but also pairs of cells, for example, CD8s specific to HIV with latent CD4 cells, to understand how CD8s can kill the CD4s immediately after activation. So we can look at things like exhaustion and other potential medical events of interest. And it allows for also screening of pharmaceutical and immunological multiplexing with cytokines and other types of tools. So I'd like to thank the people who have worked with me on this. I didn't put some other of the names there as well, but Dave Murdoch, also John Yee and Zach, and we've had conversations with Kent and Cliburn. I have collaborators in Korea, Cho-Hee Kim and his colleagues, and these are the students who have worked on this project. And I'd be happy to entertain any questions. Yes?
That's a really beautiful system. Have you looked at the interactions between the cells in your system? You said initially that there wasn't a lot of biologic effect, but what about adherent cells that might not have the same response?
We have not formed pairs of cells yet in our system. This is a difficult thing because we're still working out some of the problems with cells sticking to the substrate, and we're also developing hydrodynamic shields to prevent them from escaping the spots that they have when there's minor fluid disturbance. I anticipate that we will be able to answer that question in 6 to 12 months.
So another comment, I agree that in vivo latency is challenging because you can't identify the latent cell, but your ability to be able to recognize the transcriptional patterns associated with an activation event, I think gives you tremendous tools then, and expression patterns can go back and look, maybe not in this system, but in cells and vivo. So I think it's great.
This is a great idea. I hope it will work. The problem is that signal-to-noise ratio in vivo cells, unless you're going to analyze every single cell, is going to be a mixture of all these activation events. And it could be very expensive to do. But if you could do that, then you would learn a lot.